if this slide advances.
Huh. Now they tell us. Okay, go for it. Sorry, Cori.
That's okay.
Yeah.
All right. So yeah, thanks for the invitation, you guys. I really never really know how much to say in these talks because my husband always laughs and says once my bat switch gets turned on, it's hard to turn it off. So it's always hard for me to really restrict what it is that I want to tell you because I could go on and on about things. But today I am trying to really just focus on giving you an update on white nose syndrome, where we're at with research, some of the new things that have kind of either been published in the last little while or that I've heard about at conferences or things that we're doing through my program with Wildlife Conservation Society Canada. I'll do a little bit on climate change at the end, I think just kind of interest sake. And kind of wrapped up in all of this is really our kind of ongoing struggle with trying to understand the winter ecology for bats. And that's something I won't go into a lot on this, but that's something that, that we could talk about in the questions if there's more interest about just some of the general things. As I mentioned, like bats don't eat harvestmen in the winter. We were just talking about that before the recording started. And these are some of the things, that we're trying to understand about bats in winter, but we're not learning a lot because it's pretty tough to study them in the winter, of course. So, but gradually we are learning more through programs like BatCaver, for example, and that's thank you to you guys. And so I will talk about BatCaver today and definitely talk about some of the stuff that BatCaver has contributed to. Oh, it is going to work great. Okay, so before I begin, shameless promotion of the book it's actually like literally 5 minutes before this talk, I'm working on the proofs that are due at 9 a.m. Tomorrow, and then it goes to the press. So this is through the Royal BC Museum. It is the second edition. So the first edition was done many years ago by David Nagorson and Mark Brigham. And so those two plus myself and Jared Hobbs have put together the second edition, which will be out in March. And it's really kind of everything that we know about bats in British Columbia. But there is actually, because of my background, there is some stuff about Alberta in there. And then we just talk about a lot of things that are just sort of useful for understanding bats in general, so more on a North American context as well. So I'm not going to go into much that's in this book today, but I will kind of highlight a few of the things along the way if they come up. So, and we can talk about that later too, if there's any questions specifically about it. But one of the things that was something we pointed out in the book, because there's a lot of photos, is that we started this book before COVID. And since COVID, things have changed for how we do bat work. And so I will give you just sort of an update on that. And then I'll dive into the update on white nose syndrome. Then that kind of leads into where we are with what we call our probiotic project. And that's a way that we are hopefully going to try to reduce some of the mortality associated with that disease as it spreads further in the West. Then an overview of acoustic monitoring. Again, I won't go into a ton of that. I will dive into sort of what's going on with BatCaver, and we can talk about more about that in the question period if there's any questions. And then climate change. So I guess models more or less concerns and how that relates a little bit well. And then we'll just sort of throughout this sort of talk about the knowledge gaps and further work, and particularly about BatCaver. I'm not really sure where we're going with BatCaver, honestly. And so I would like to tell you what we've done so far. Many of you have been involved in that. And then maybe there'll be a brainstorming session later as to what some of the things that we could do in the future. Okay, so to start off, COVID. I'm not going to dive into too much about where it could come from. Yes, it does seem to have stemmed from bats, but not North American bats. And mostly the reason for that is there is a difference between beta coronaviruses and alpha coronaviruses. And what COVID-19 is a beta coronavirus. They are found in bats around the world, but in North America, we just have so far only found alpha viruses. Beta viruses are the ones that are typically associated more with dangerous human disease. So you might kind of wonder what's going on here. Everybody's wearing masks. It is definitely a concern that we do not want to spread the COVID-19 virus, the SARS-CoV-2, to the bats. And so this is kind of, a precaution that we've been taking since the pandemic started. For a while there was a moratorium on bat capture completely. And then we've been kind of gradually going back to doing it on high priority projects, but always wearing masks. And whenever we're close to a bat, then we make sure that there's no way that we're breathing on that bat, essentially. And that's because we don't know the susceptibility. There's been a little research done so far that has pointed at a few species, like pallid bat, for example, that they do think is susceptible and could pick up this virus. We don't know what the virus would do to the bats, if anything, but it could also become a reservoir then, so that North American bats might have this beta coronavirus, for example, and who knows what could be the ramifications of that. So we are taking all precautions. And for the foreseeable future, then we wear masks now to handle bats, even if we're vaccinated. All right, so new knowledge. What have we learned about the PD fungus? So there's just a couple of things, I guess, that have come up over recent years. Not all of this is necessarily published. I just thought I would share that the USGS communicated through some of the White Nose conferences that they've been able to track PD spores, the spores that cause the fungus, cause the white nose syndrome disease. And they've kind of tracked that they can live for a fairly long time and stay viable, to say, on bats, like in their fur. Bats do groom a lot, and those spores will enter in through their digestive tract. So over time, those spores are likely to be lost. But it could be like 2 to 20 weeks, depending on the body temperature of the bat and depending on how much grooming they're doing, which means there's this potential for long distance dispersal of the spores. And of course, that's probably what we've been seeing in the East over the last decade is mostly this bat to bat transfer of spores. And now understanding how long they could potentially stay on a bat puts that a little bit more into perspective. One of the things that was uncertain for a while is whether the spores of PD, when they land into cave sediments, could they grow, or would they just persist? And some of the research lately has suggested that they really don't grow in most soils. When these spores do land in cave sediments, they will persist, but they're not necessarily going to grow. And that's mainly, it seems like, because of the microflora that's already there, that for a variety of reasons, whether it's through metabolites being produced by the bacteria or fungi, or just competition. There's a whole bunch that they don't know about that, but they have found that this is to be the case, that we're not looking at growth. They did take some soils and sterilize them, and some of those soils then didn't grow the PD, but most of them did. So once you sterilize the soils, it looks like whatever was in there that was competing or inhibiting the PD fungus is taken out through sterilization. So those are the two main new things. Now, what that means is we're looking at spread maybe a little differently, and what are the chances that it's spreading through cave sediments versus bats themselves? Well, looks like what we've all kind of put together over the years is that the bats are mostly spreading it. Now, where are we going to pick up on these PD spores? Understanding that spread pattern has been actually really challenging, and we've got more evidence these days to suggest that we haven't really documented the spread in a way that we thought we had, and I'll get to that in a second. But first of all, just point out the usefulness of bridges that has become very obvious over the last year or so. In Montana, during the pandemic, they weren't handling bats, so they actually just started to look at where bats hung out, and in particular night roofs of bridges, and they did find the PD fungus under some of the bridges, and that allowed Montana to sort of know that they had the fungus in some of their eastern counties. This past summer, our program, actually the Alberta portion of WCS Canada Bat Program run by Cori Lausen, went through and sampled bridges in both Alberta and Saskatchewan. And he did find PD in Saskatchewan. So that was the first record of that. And so the new map, kind of as of last month, after all of the results were confirmed, has it in Saskatchewan and basically three locations, creeping ever so closely to Alberta, to the Cypress Hills area. This is a little bit surprising because these bridge samples, of course, were taken middle of the summer. So that means that PD is persisting for a long time in these kind of cool, moist conditions that can exist under bridges where the guano just clings to the concrete and stays out of the sun. So this is a good way to probably continue to find PD, and maybe a better approach to trying to understand its spread than what we've been doing up to this point, especially in BC here. We're just waiting for people to turn in bats. And that's largely the way that we've been looking for the fungus over most of the years, until very recently when they started to also look at swabs of bats. So that, I don't think this map is actually updated yet on the whitenosesyndrome.org site. It is on the Canadian Wildlife Cooperative site. This one then is the map kind of showing this bigger picture, what's going on in North America. You can see then that the Saskatchewan records aren't on there yet, but you can see Washington. And of course, Washington, the spread there has been going on since 2016. More counties. There's been undocumented cases, like people showing pictures and phoning, and unfortunately, those cases, a lot of them do look like they might be white nose. One in particular over on Orcas Island was unconfirmed, but it had a lot of signs that suggested it was white nose syndrome in a bat, but there was no sample, no carcass, unfortunately. So that's only about 10 kilometers from Victoria, which means, just as we expected, it is creeping closer. And the tricky part is actually being able to confirm that we have the fungus. So far, still no confirmed cases in British Columbia. In Washington, just last year, kind of during the pandemic, then again, it was hard because nobody could really handle bats, but they were going and watching bats at some of the colonies where the PD infections are known, and they were starting to see that. So the mortality is definitely ramping up in Washington. And so this observation of dying bats is certainly something that we expected. And yet the fungus has been there, we thought, since at least 2016. So it's kind of slow to come, and I'll talk about that in a second as to why we might be thinking it's slower. Okay, so what else is new? How do we find PD fungus? Well, yeah, there's bridges and there's the typical PCR tests, but there's some interesting stuff that's out there that's kind of new and coming online. eNose is one of these really interesting new kind of biosensor type of tools that is set up to analyze volatile organic compounds, so VOCs, and it's able to pick up on very small particles, essentially molecules of chemicals that are associated with certain organisms. So this task can find PD potentially. It's also able to tell species apart. So it's sort of in its early stages. And this publication is just one that starts to outline what are the potential uses of eNose. And certainly detection of PD and actually being able to tell different bat species apart is really, because you could almost just go into a cave environment, for example, and sense the molecules in the air and be able to tell if it's a PD positive area and what bat species are likely hibernating in there. In fact, this isn't in the publication, but they did mention in a conference, this lab, Dottie, they have also found that they can tell pesticides that bats are being exposed to by the bats breathing, because they've eaten these insects that have the pesticide, and they breathe that pesticide. And so they can actually start to pick up on those kind of chemicals as well, which is really interesting. And probably something that will be used in the future. So eDNA, okay, similar concept, looking for small molecules, but obviously it's out there looking for actual DNA. It's a tool with a lot of promise. Has been used for mostly aquatics in the past, but now looking at how it could be used in other ways. There is this paper, and then there's some other kind of unpublished stuff that's really looking at eDNA being used to detect bat species and also to detect PD. So one could, and in fact, in this paper, they were taking water samples running out of caves. They were also just going out to rivers and lakes and identifying the species of bats that are around because of the guano that presumably would fall into the water source. But they've also been able to do just kind of from sediments in the cave, be able to tell what species of bats are in there. And this part is really cool, is air. And so this is kind of allude to the fact that they're working more on this air component of eDNA, but they have tried it on an enclosure, for example, and it works. So really, they just keep filtering air and getting all of the DNA that's floating around in the air and being able to tell then again PD and bat species. So these are really sensitive tests, and far more sensitive than what we've had up to this point for looking for PD fungus. And it might be that we've had insensitive tests because this was a new fungus and we didn't know how to look for it. And so for a number of years, the USGS was sort of leading the way on trying to detect this fungus and develop tools to detect it. And in the beginning, they were not all that sensitive. And maybe that's part of the reason we kind of saw a spread that we thought we did, and why now there might be other explanations for how the fungus actually spread. And that's what brings me into kind of what has been seen as sort of a potentially slower spread in the West. As I said, the fungus was detected in 2016 in Washington, we know that, but was that when the fungus actually arrived? And again, we thought the fungus arrived in New York in 2006. Bats started dying in 2007, and it looked like it sort of radiated out from there, and then the spread continued, and in 2005 it showed up in Ontario, for example, or Quebec, and then spread from there. So the recent research by Marilyn Rusnick and her student Thapa, Bhaskar Thapa, who actually published these two papers here, looked into what's called a Partidi virus. And if you haven't heard of that, it's actually a virus that infects the PD fungus, the Pseudogymnostus destructans. So the virus infects the fungus. And it is actually, interestingly enough, the viral infection which allows PD to cause disease, to actually be have its virility. So the fungus traveling around North America travels with this fairly fast mutating virus. And by looking at the virus then, and looking at the genetic sequences and how that virus has been mutating over time, has allowed them to map out where they think the original introduction was, and then the spread pattern from there. So this is all published. People are still debating, because of course this is always a question as to, do our models of how these genetic mutations happen, are they actually showing us what we think they're showing us? But based on what has come out of this research, the actual introduction into North America, it looks like probably was in Connecticut, and probably back in 1999, or possibly even as early as 1996. And then it spread like to Alabama and South Carolina, and it sort of spread around in a different pattern than what it was discovered as we went along since 2006. This research also shows that it was in New Brunswick in 2005, so 5 years earlier than it was thought to be in Canada, and it was in New Brunswick. And then basically as of 2014, something transported it over to Washington. So 2 years, they figure 2 years earlier, than when it was actually discovered in Washington. And it looks like it was a point source introduction from Kentucky. So that's kind of, it's changing the way we think about the spread. It doesn't mean that this is necessarily the right, the way things actually happen, but at least we are getting a better picture of some of the things that might have contributed to what we are now seeing in the West as somewhat of a slower spread, because in reality, if in fact the fungus showed up earlier in North America, and if the spread pattern has been the way that Thapa et al. Has been suggesting, that means there's about a 6 to 8 year time lag in which the fungus arrives, is not necessarily detected, and then we ultimately end up with bats being discovered dying of the disease. So that's about where we would sit here in the West if the introduction was in 2014. Last year was the first year that they were starting to see these mortalities of bats in some of the kind of ground zero areas of Washington. So that sort of lines up with what we'd expect. And now we're watching, this what we consider kind of to be a slow spread, but it's probably the same spread speed, or very similar anyways, as to what actually did happen over in the East. But having said that, there are other contributing factors in the West that are likely contributing to this slower spread of white nose. It could be that our bats are just doing something different. And this has been suggested for a number of years now, and certainly hypothesized or sort of compiled together by Ted Weller in his publication 2018, which looked at what do we know about bats in the West in the winter. And the answer really is, well, not much. But we do see some aggregations of some species. There are always small aggregations. I mean, the largest hibernaculum for little browns, for example, that we know of in the West is up in Northwest Territories, up by Fort Smith. And that is 3,000 little browns. And that's huge for the West, but that's not huge for the East. And so we definitely have very different winter ecological pattern of what's going on with bats here in the West. We don't necessarily know where they hibernate, but they're not hibernating in ways that we would have expected based on what we see out of the East. And could that be influencing how white nose is spreading? Yes, it certainly could. Okay, how are we mitigating white nose syndrome? So there's a number of things that are happening now with Wildlife Conservation Society Canada. I'm going to end with that one. I mean, talking about the probiotic, but there are a few other things I would just mention that other people are doing because they're not necessarily published, and there's some mixed results out there. So what's called fat bats, Bat Conservation International and others have been looking at taking this concept that we know that bats are attracted, so that insects are attracted to lights, and what if we put lights out in front of these hibernacula and attracted a whole bunch of insects, and so when the bats arrive, there's this buffet for them to feed on right before they go into hibernation. That's sort of some ongoing work right now over in the East. Hot boxes, an idea that came out of University of Winnipeg with Craig Willis, basically says, hey, let's look at whether we could help these females when they emerge from hibernation. If they have, just barely made it through the winter surviving white nose, how are they going to possibly have a pup? Could we give them a nice warm environment in which to help develop their fetus and heal their wings? And that looks like that's been successful in many regards. And they're still working on it in terms of like field data, but they do have a paper with some preliminary data that showed that it should be very beneficial to females, and they do seem to select these warm boxes. Now, the Canadian Wildlife Health Cooperative, they have decided to change what they used to call our white nose response plan in Canada. That just changed over this fall to be a One Health type of program, Bat Health. And so Bat Health is really kind of this big picture of everything, not just white nose syndrome. So how can we help bats? How can we reduce threats to build resiliency into populations and start to think about all of the other things that bats need to either keep them from being impacted so much or at least help them recover. And so that's the approach that we're, a lot of us are starting to focus on, is we might not be able to do something about white nose syndrome on a big scale to biologically be significant, but we can start to look at all the cumulative effects and see if we can reduce some of those. Now, having said that, for the West, there are two different alternative mitigation strategies that are in the works and might still play a role in reducing the mortality caused by white nose syndrome. One is there's a publication by Roki et al. And actually it was 2019, I guess, of some preliminary data. This research on the PD vaccine continues. In fact, her team was out into Washington in the fall and vaccinating bats at some of these caves and mines where they could capture bats. They are putting, they feed the vaccine to the bats, and they're putting on in pit tagged readers, and so they can hopefully see how well the vaccine does, if it actually builds immunity in the bats, and then the bats are more likely to survive white nose syndrome. So that's all sort of ongoing, and we're all waiting to see how that works out. Vaccine on fungi is actually a really difficult thing. And then there's this probiotic approach. So Joe Hoyt in 2019 had a paper showing that if he sprayed Pseudomonas fluorescens, which is a soil bacteria basically, onto bats in the winter, would they do better at surviving white nose? And yes, they did. And so since then there's been more examination of the microbiome of bats, and people are really just diving into this. In fact, this is a collaborative project I'm doing with McMaster right now that might start this summer, is really getting a better picture of the microbiomes of bats across the West and comparing to the East, because it does look like the other microorganisms on the bat's wing can be a big predictor as to how susceptible they are to white nose. And therefore, it might be then that this is the route to go to try to reduce mortality. Or maybe it's a combination of a vaccine and probiotic. And if they each have, let's say, 30% success, then maybe used together would be more effective. So these are the sorts of things that are happening. I'll just give you a little more information about the probiotic cocktail that we have developed, in case you hadn't heard of it. We are couple of years in now. This has been developed. It's been tested in a captive trial, several captive trials, I should say, and it is being field tested now. But the way we came up with this was we sampled 13 species of bats, several hundred, a little more than 300, and then with McMaster and Thompson Rivers Universities developed a probiotic made of 4 bacteria that are naturally found on bats' wings, but do work together to inhibit the PD fungus. So it's considered a prophylactic. So this is kind of unique in the West. We're not going out and trying to find bats hibernating. There's just not enough of them to make it relevant to actually go out and try to save bats in hibernacula. But we can focus on kind of preventing the disease by building up within the wing microbiome these anti-PD microbes. And then these females, these are the ones we'd like to target because we know where large groups of females hang out in the summer, i.e. in buildings and bat boxes around the West. We are definitely, it's definitely an advantage to be able to focus on the females, because we know that in the hibernacula of the ones that have been found, a lot of times there is a male bias. So again, by focusing on the summer, we're actually focusing on females that are going to be more important to recover these populations or keep these populations from being extirpated. And so we are looking at community bat programs, for example, like the one in Alberta and the one in British Columbia, so that we can potentially upscale this in the near future and literally turn it into a citizen science type of project where we can have these good beneficial microbes essentially in a very easy to disperse box that goes out to people, and it consists of a spray can of air, and you can actually see the picture here down in the bottom. Spray can of air and a little tube which has the microbes in there and a little bit of powdered clay, and it just gets sprayed up into the bat box. It's really just non-pathogenic, safe microbes that would naturally be in the soil, and some bats already have these. but most don't. So this is sort of like that Robin Hood approach of trying to get it onto more bats, especially ones that roost on plywood, with the assumption that maybe they have not come across these natural microbes in the soil because of the fact they're not roosting against natural surfaces when they're in our buildings and in our bat boxes. I guess I'll just point out the very end there. I did just make a point there of saying we think those bacteria are actually found outside of BC, even though we have actually sourced them from within BC. We've just started looking at some of these genetic databases, for the better lack of a word. They're not really databases, but they're a bunch of genetic sequences out there that we can start to look for the bacteria that we found against ones that other people have found. And at least 3 of our microbes are showing up in soils across Canada. So it might be that our probiotic could be used outside of British Columbia, but right now we're still just keeping it within the Pacific Northwest. I won't spend much time on this slide, but I just wanted to kind of acknowledge that there's a large group of people that help make decisions on this project, because it seems like we're just always almost making decisions every time we meet, because there's just new things that are coming up. And as we find out one thing, it leads to another. And we're all trying to make decisions that will help us approach this project in a way that we can hopefully upscale and use it outside of our current study areas. And those current study areas are in Southwest BC. And we've just gone to Washington. Now in 2021, we just picked out study sites, and it'll be in 2022. Hopefully we'll be providing the probiotic down there for the Washington Fish and Wildlife to put into some of the bat boxes that we're including in this study. We just have to secure all of the permits, but the funding is now in place. Okay, so switching gears a little, and I'll come back to these ideas in a second here, but I just wanted to just very briefly talk about acoustic monitoring, because we really don't have a good way to tell how bats are going to be impacted by white nose syndrome. They're already being impacted by so many other threats. And so having a way of understanding bat populations has been difficult, but we are doing the North American Bat Monitoring Program. And then there's also BatCaver, of course, and these are the programs that have over the past few years helped us sort of figure out like who's where, and do we have large numbers of bats in certain species in certain areas. So BatCaver, you're all familiar with that. And we have appreciated all of the help that the cavers have provided. You guys have given us a lot of data of what the underground environment looks like. So even though in BC we didn't find a lot of bats, in Alberta there were new hibernacula discovered, but over a very large area we have started to get a good sense of what are the microclimates like. And that has led to, and I apologize, I'm going to show a few graphs, but I'm not a modeler, and most of the papers that have come out with using some of the data that was collected through BatCaver are very intense survivorship models, and based on respirometry data in addition to what the microclimates are. But I just wanted to let you see that these data have already been useful in trying to understand what our bats are doing in the winter, and which ones are susceptible based on what we know about where they are and what kind of conditions they're roosting in. So just very briefly in this one, this paper came out in 2020 on survivorship models, and they just looked at the energy consumed during hibernation and in relation to how well a fungus grows. So is it cold? Is it moist? Where they're hibernating, what are the chances that the fungus is going to grow well or not grow well? So just really briefly on the top here, the temperature, the warmer the temperature, okay, the bats are not doing well. So this is probability survivorship. So here you can see like Big Brown, this is Big Brown town, so it's big ear bat. They're going to do well. And we know that they do well in the east because they're using these cooler temperatures. Okay, down here, water vapor deficit just simply means how dry it is. So the Townsend's bat, the Big Brown and the Cave Myotis and the tricolor bat and the Western small-footed, these guys are all sort of choosing drier roosts. Okay, and these ones here, again, you know, they are sitting with higher survivability, but not all. So again, this is just where these models are trying to now figure out, like, if we combine all of these things together, who's likely to survive and who isn't. And the last thing that they're including in these models is body mass. And so the larger bats are the ones that are less impacted. So bats like Townsend's big-eared, big brown, cave biologist, which we don't have here. And then the most impacted would be the smaller bats. So some of these things are a little bit science of the obvious, but now we're at least figuring out where we could plunk in some of these numbers and come up with a survival probability. Also, BatCaver data has fed in a lot of information then over the years about what is our winter length. So, we've got winter length being defined all over the continent, but in particular, as we go further north, it was really not certain as to how long is winter, how long do bats actually hibernate for. They don't seem to hibernate for as long as one would expect based on when there is snow on the ground. So temperatures have within the microclimates, temperatures within the roosts that have come out of places like Catamount Cave and Self-Slave Hibernaculum in Northwest Territories, those have helped to predict whether little browns in those areas, for example, are going to survive white nose syndrome. And I won't go into the details here, but I will just highlight uninfected, i.e. healthy bats, infected with white nose. This number here too means that this is how much body fat they would have left at the end of the winter if they weren't infected versus if they are infected. And this A here represents like Catamount Cave type conditions, 4 degrees and 98% humidity. And then B represents more like what you'd find up in the Northwest Territories, 2% and 100% humidity. What this means is that bats will still have a little bit of fat remaining at the end of the winter. And so it might be that these northern areas do still represent a bit of a refugia for white nose syndrome. For the longest time, we weren't quite sure, but it's also not clear if this is, if there's other factors that aren't being considered right now. But so far, it does seem like there's hope for the further north bats. And there's just more, I won't go into all these maps, but if you were interested, I can either pass along those data or we can talk about them later. Okay, and then microclimate data has also contributed to predictions about where we find bats. Okay, so where, if we start to model where bats are based on what types of temperatures you would expect in the underground environments, what the BatCaver data has helped, of course, you can see like data collected all over Western Canada. It's helped predict where bats will be based on what the underground microclimates actually are in relation to what we can predict from the mean surface temperatures. Those mean annual surface temperatures is generally used to predict underground temperatures, but there is a model put together then in this paper, McClure et al., that has looked at how can we really get a better sense of what the underground temperatures are like other than just using mast. And some of the things that came out of that work is that mines tend to be warmer and caves tend to be cooler. And this is kind of obvious, I guess, as you get deeper into the feature. It's more stable, but it's also a little warmer. And there were other things that were put into the model as well. Overall, then, these things have been plotted out to try to understand underground temperatures in relation to bat hibernation. So that's just, I won't go into that one. That's just a bunch of cave versus mine, but I will show you this, which is kind of interesting because they broke it up into Alberta versus Texas. And what this means is here, this pink is what we'd expect based on the mean annual surface temperatures. And this is caves in green, and this is blue in for mines. And then the gray represents where bats would typically hibernate. And this just suggests that in Alberta, there's a large portion of the mines that could potentially be good for hibernating bats. And then, of course, there's some caves. Now, I actually think that applies actually more to British Columbia, because certainly we're finding a lot of caves bats in mines in British Columbia and a lot less in caves. So this might also apply over here. And finally, the little brown in particular. The little brown then uses these temperatures to hibernate in, the gray, in which it can take advantage of both cave and mines. But what's interesting is that it's the caves that provide the cooler environments. And so it might be that little brown is more likely to survive in caves with white nose syndrome than mines in white nose syndrome, with white nose syndrome. Okay, so the last kind of topic I wanted to talk about with BatCaver is really about critical habitat. And critical habitat applies to the two federally endangered species, little browns and northern myotis. We know that they both occur in Alberta, and we actually both in British Columbia as well. We have been able to find through BatCaver, with, because of you guys' work, been able to find new hibernacula in Alberta, and many of them contain both of these species. That's not been the case in British Columbia. We still actually have no identified critical habitat for these bats, so we don't know where they overwinter. There is speculation as to where they overwinter, and that's because of just uncertainty of species. And people like Martin done a great job of going in and finding where we have bats. The tricky part is even bat biologists, we just can't tell them apart, right? It's just the little brown from the Yuma myotis, and unfortunately, the little brown is the one that's endangered and listed, and the Yuma is not. And yet, you look at them and they're almost impossible to tell apart. You really need to get an acoustic recording from them. You're not going to get that when they're hibernating, or you need a genetic sample. And again, that's something you're not necessarily going to get either because of the disturbance to then try to genetically sample a bat. So this is why, unfortunately, we sit with still no critical habitat in BC. But maybe we should start looking at moving forward and trying to fix that a little bit. So, is the next step trying to find some of, go into some of these caves that have been identified through BatCaver with bats in them, and do we actually try to get some samples from some of these bats and finally figure out where we have these species. It might also be that they're using a lot of rock crevices, and we may never find a lot of hibernacula for these species. We do know in Southeast Alaska, for example, they are using root wads. And so if they're doing something like that in British Columbia, especially along the coast, well, then we are unlikely to find them, or at least if we did find them through something like radio telemetry, we're not going to be able to sample them. Okay, I'm going to skip this because I've actually already talked about North American Bat Monitoring Program a little. How am I doing for time? Yeah, I'm just going to skip this. So the last kind of interesting thing I thought I would break forth is not necessarily anything to do with caves and white nose syndrome, but it is something that has been a growing concern for bats in the West and then across the world, really, is this idea that bat boxes, which are becoming very popular in recent years, might be acting in some cases as ecological sinks. They might present places where bats can't actually exist during heat waves. And if they don't have somewhere else to go, it might end up resulting in mortality. So we have had some mass mortality events in British Columbia during heat waves. This has also been seen in the world, Australia and Spain, places then that are starting to realize that we might have a problem with the use of bat boxes in that we're not providing enough choice. And we can't just assume that you put up a few bat boxes and these bats now depend on these bat boxes instead of depending on a building where they would have found a lot of different microclimates to keep them either warm or cold, depending on what they needed. But in bat boxes, they don't have that option. They're so small. So we are coming out with some BMPs that hopefully will be out early in the year, or sorry, by spring, I suppose. And I've also done some webinars, and I've also put together an article that was just released to the public in relation to COP26, and that's in the Bat Conservation Trust newsletter. They focused on bats and climate change. So that's, if you're interested in that sort of thing, there is an article that I just call the Goldilocks approach, because it is really about giving bats choices. They do need more and more choices if we're going to see these extremes in weather. Okay, the very last thing that we're working on in BC see right now, but eventually, is an alternative to bat boxes. And that is, especially in these non-urban areas, we probably shouldn't be sticking up bat boxes, which attract just certain species. In fact, most of the time it's 2 or 3 species of our potential 16 or so that could be going into these. So if we were to appeal to tree roosting bats, ones that will go up into bark or into cavities, then maybe by using something like fake bark, like this, what we call Brandon bark, or by creating wildlife snags, like literally taking a fairly large tree and topping it, and then making strategic cuts in it and ringing it so that it will be a future snag, but in the immediate future, there'll be these chainsaw slices, which we're calling like slabs and plunges, creating these little nooks and crannies for bats to hide in. And that, in theory, will attract different species or more species than what we would get with bat boxes. And that's probably a good thing, because otherwise, these bat boxes, if they're spread all over the place and we just have building roosting bats like little brown or Yuma myotis or big browns using them, then those bats might start to dominate some of our communities of bats, and we would see a lowering of the biodiversity over time. So this is where we're heading right now with some of our mitigation attempts. And again, these are our ways of trying to look at things other than directly treating white nose. What else can we do to try to build resiliency in our bat populations in the West? And I will end there. I definitely went over time, but I guess we started a little bit late too. So if there's any questions, and if you are interested in checking out anything more about the programs, obviously I'm not going through much of them today, but there is information on my website, wcsbats.ca. And my social media person always likes me to say, follow us on Twitter and Facebook. So the end.
So, Cori, it's Phil Whitfield here. Thanks so much for doing this. I'd been unplugged from the bat scene for quite some time now, and it was just an amazing update. Really appreciate that. I would hope that maybe we could, maybe go through the lines and something like that for some publications to keep people up. Working with anyone east of Winnipeg on any of this stuff, because we just discovered that supposedly the Nova Scotia government believes that the white nose syndrome was brought to Nova Scotia caves by cavers, and there aren't very many cavers in Nova Scotia, if any. And we're wondering whether they need to be made aware of the collaboration between, the bat researchers and cavers in the West, and how successful that's been, so that we don't end up with a sort of what is apparently presently a uniform ban on caving anywhere in Nova Scotia.
Yeah, I must admit my program doesn't go past Saskatchewan. Not for, just reason, I suppose, we're here. Cori Lausen from Alberta one, and I run the BC one. So we haven't, but that's not to say that, we can't, have these conversations with key people. For example, Jordi Seegers is the Canadian Wildlife Health Cooperative White Nose Syndrome Coordinator. Now he's the bat health coordinator. And he's out on PEI, so he might be a good person to bring into these conversations. I know you're amazingly busy,
but maybe I'll touch base with you sometime after this session here, and maybe get some contacts and see what we can do. Because the other thing we thought was it would be really useful just to get your endorsement of the fact that cavers are not necessarily the enemy and might even be allies in a lot of this stuff. For some of those governments who really just think that shutting down a cave solves all their problems.
Mm-hmm. Yeah, it's been an interesting struggle from day one, hasn't it?
Yes. Oh, it sure has. Yeah, and it continues, unfortunately.
It does, and yet continues with not a lot of evidence. So I would be interested to hear, what the evidence is, that they're pointing out for out there. Yeah.
Great. Okay. Well, I'll be in touch. Thanks very much. A wonderful presentation. Thanks again.
Oh, you're welcome. Thanks.
Hi, Cori. Jason Ray here.
Yeah.
I was just wondering, you were talking about the beginning, or closer to the beginning, about how it was originally introduced through, you think, Connecticut. What do you think was the source of that? Because one time I heard something about some guys trying to grow some cave cheese, and they brought some soil from France. And is there any truth to that?
I don't know. There's so many rumors out there, unfortunately. And yeah, and not, I don't even know that they're necessarily rumors per se. There's actually, because that whole cheese curing thing and bringing dirt from France into that Albany cave, I think did happen. Whether that's what introduced the fungus, though, I mean, that's the part that is probably at this point just, a leap of faith. But I think there's a lot more to look at now. And this paper that came out by Thapa et al. Just, it's ruffled some feathers anyways, and we don't know. I don't think we can point to what the real story is, and maybe we will never know. But I think it's at least opened up this door to this idea that we think we saw the spread, but maybe that's not, maybe that's not really what happened. I mean, what are the chances of discovering it in the cave where it was first introduced? It's actually kind of low when you start to think about it. So it seems like that introduction was not necessarily in New York, but do we know how that came over? No. And was it people? Again, we don't know. Could it be spread by bats, catching rides? Yeah, it could, because that's documented a lot. Like there's several review papers out there now that just point to countless cases of bats being transported across the ocean, for example. I mean, we had that happen in Vancouver, Port of Vancouver. Big ship from China pulled in years ago. Maybe 2009 or so. And I was contacted, and they had a whole flock of bats that were flying around inside this cargo ship, and it sat open overnight. And they showed me a picture of this, and I'm like, that is not North American. That bat's definitely not North American. And so a whole bunch of us here in Canada were contacted bat people, and we're like, yeah, no, that's definitely not North American. But they fumigated the whole thing, but they never did find any more bodies of those bats. So who knows, right? I mean, these things happen all the time, that bats get moved around, and not just by long distance transport trucks within our country, within our continent, overseas as well. So I don't know that we will ever know how it got introduced personally, but there's definitely, I think, this whole idea of the Partiti virus and being able to use that as a tool to try to understand this more. I think that has opened up a whole new door to our understanding of the spread of this fungus.
Thank you for answering that. One other question I had was about those PD spores, and you said they can persist in the cave, in the medium. Is that something that we as cavers could be transferring between caves?
Mm-hmm. Yeah, so you can transfer the spores for sure. So those spores, they build up in the sediments, and then if you were to get the spores up in the mud of your boots, for example, if those boots weren't decontaminated, and you took some of that mud over to another cave or like uninfected, infected. I mean, obviously these are important things to think about. Like, are you leaving an area that has PD and going to an area that that doesn't? I mean, the decontamination protocol would be really, really important for that, because you would potentially transfer spores. The harder part is to understand what happens to those spores, because if they don't grow, then the number of spores that you've taken might be the number of spores that stay there, right? So if there's a lot of cavers carrying a lot of spores, then sure, there'll be a lot of spores in that sediment. But based on the research that's come out of the USGS, it looks like it's not like you're suddenly going to have a bunch of spores that are causing germination and then growing of PD and more spore production in those sediments. That would sort of require that a bat pick up the spores from the sediments and then and then those spores could germinate on the bat, grow in the wing tissue, produce more spores, and voila, now you've got the disease cycle. Could it happen more than just boot sole? Like, could it happen like you are moving around and you pick up a bunch of spores on your jacket, and then you go into another cave and you're crawling around and those spores then move from your jacket onto a wall where a bat would roost. So that is a more dangerous situation, because then that spore sits there. It's not going to go away. It's going to still be viable for a very, very long time, because these fungal spores can stay dormant and still be viable for a very long time. So I think that's the stuff we have to, be really cognizant of.
Thank you.
Cori.
Hey, Martin.
Yeah, I found that really interesting what you're talking about, the virus fungus connection. Do you know any more about that? Can you speak more about it? Because it's the first I've heard of it.
Well, if I had freshly read the papers, I might be able to tell you a little bit more. I'm trying to remember back now. Yeah, I mean, I could send you those papers if you want, but so the virus was discovered back in 2016. And I mean, it's a very interesting relationship between the virus and the fungus, because without that virus, the fungus is not deadly to bats. So part of what people have been brainstorming over the time since that has been discovered is maybe we should be looking at somehow disarming the virus. If you can disarm the virus, then the fungus could be there but not be virulent. But I've heard nothing on that front. I mean, that was sort of hypothesized in the beginning. Hey, maybe that's a possibility. It might be happening. A lot of this research tends to happen sort of behind closed doors until they've kind of come up with something and then they present. So.
Yeah, no, it's really interesting. I just never heard anything about that before. And also, since I've been studying bats, I've always noticed funguses of various kinds on bats. You get the white fungus and the green fungus, and certainly they dominate if the bat dies and hangs off the wall after it's dead, and you'll see it cover the bat eventually. But they seem to live with a lot of funguses as it is. But
they really do. They really do. It's amazing. Yeah, like these bats. Surprising that they don't mold up more from just any kind of fungus, really. Yeah, but especially because when they were hibernating, of course, like their immune systems are largely shut down, and so this is something that that over time it seems like they're trying to jumpstart their immune system over the course of the hibernation period and fight off this PD fungus. But none of the other fungi try to, grow on their skin. And so I guess they don't have a problem with any of those other ones. But yeah, the minute they die, they start to get covered in fungi, and you realize how much fungus is actually in these cave environments. It's pretty amazing. Very colorful stuff too.
Yeah. So when you think about it, they have probably developed a lot of survival strategies for living with these funguses. And I guess every once in a while something comes along that they can't deal with, and hopefully they will be able to figure it out.
Yeah, totally.
Mm-hmm. Yeah.
Hey, Cori, it's Kirk.
Oh, hey, Kirk. How are you?
I'm good. I noticed you skipped by a slide that I think was showing it showed the bat species, and it looked like a graph of relative health or something of the bats, and I was interested
Go back to it.
here. I'm wondering if you could.
Yes, if I had noticed you were on, Kirk, I would have stopped on that slide. I just looked at the time and thought, oh, I better wrap up this one here, right?
Yeah.
Okay. Yeah. So this is a report that. So I'm on the like Continental Program committee, and this is long overdue. This State of the Bats report. The first one we'd hope to have it out actually back last spring, but it will be out this spring, if not like kind of early in 2022. And this is just one of a number of graphs that shows overall what's happening to bat species that we've been tracking them through NABAT. So the North American Bat Monitoring Program, which of course, Kirk, you're obviously very involved in, and BC Parks in general has been monitoring in a number of grid cells over the last few years. So as we get more and more data, this is largely based on the 5-year data set, but this will be repeated every year as more and more cells come up to that kind of 5-year data. I'm going to go back one. Yeah, here. So you can kind of see like a lot of monitoring is happening continentally. And just for your sake, Kirk, I would say that most of these cells, the data that we're getting is occupancy, and there's been less done with relative abundance. So based on occupancy data, kind of where bats are winking in or out, or rare versus common in certain grid cells, has allowed kind of these larger pictures to be painted. And this is just of all bats combined. So this is just like one out of like 30 maybe graphs that are in there. But I just kind of picked this one because it had Alberta and BC on it. But you can see the changes. So this one here, the total 3-year, is actually the more reliable one, because our grid cells in particular, like back in British Columbia, like the ones especially that have been contributed by BC Parks, only go to 3 years. But what they did was they used statistical modeling to kind of figure out, going back as far as 9 years, what it probably looked like. This may not make the final report, FYI. And so, and because it's not out, and I know this is being recorded, I do want to clarify that these two here, the 7-year and the 9-year, might not be shown. But regardless, this one here, up to 3 years or maybe even to 5 years, you can start to see, like, in places where the red means the bats have been declining overall, and then the blue suggests that they're stable. So Alberta's stable population, and British Columbia has a stable population. But these other places, which of course do correspond with eastern locations, are showing declines across all bats. So, and then there's some species specific trends that we will be releasing in that as well. We're not doing all of the species, but the ones that there's the most data on will be done this year, and then more species will be done in following years.
Yeah, that's interesting. So some of they're still considered, some are still blue, even though their mean is in the
Yeah, that's because, sorry, I got off that, but yeah, that's because the error bars they cross 0. So if 0 was included within the error bars, then it's considered a non-significant result. And
okay. Mm-hmm. And do you know for the bat box heat study that Susan's doing, is that going to be? I haven't heard anything in some time. Anyway.
Mm-hmm. Yeah. So Susan Dolk, just for the rest of you, my master's student with Cori Lausen at Thompson Rivers University. She is working on looking at comparing the kind of the health and the reproductive status of bats that use largely bat boxes to those that use buildings. And then some of her data is contributing to the best management practices that we're putting out for bat boxes. And so she did a field season in 2019, couldn't do it in 2020 because of COVID, finished up this past summer. And so she is a year behind. So she's just analyzing her data now and hoping to defend in late spring. So nothing really new to talk about there yet. Yeah.
Right. Okay. And so what else do you want us cavers to do?
What do you want to do? I must say here in BC, I just feel like we haven't nailed it yet. You guys found what seems promising up in the Northeast, right? In the Dawson Creek area there, working with Inga Jean Hansen and Brian Patterson. And I just wonder if any of that is worth trying to access in the winter. There was also Fang Cave right down by Prince George. Like there's been a few caves, and you guys will probably remember better than me, that had bad activity, and especially had bad activity at about 40 kilohertz. And, that's potential little brown or northern myotis if it's in the north part of the province, obviously. But those are the ones I think have the highest priority. Now, Martin went and did get into one of the caves at a 40K myotis. Martin, you'll have to refresh my memory, but I know you did that on Vancouver Island, but it ended up being long-leggeds, and that's the other species that will produce a 40 kilohertz call, and that you have seen hibernating in Vancouver Island too. So there's probably still caves that, if they were accessible in the winter, would be worth sampling, be worth trying to figure out, and we'd have to get a genetic sample and just finally say, yes, we can point to a dot on the map and say, we know that species is hibernating here, and especially for the federally endangered ones, because then we'd actually be able to say for sure we have critical habitat. And maybe it's not worth pointing at a map in one location. I don't know. Biologically speaking, maybe it doesn't matter, but maybe policy-wise it does. I don't know. To be able to say something concrete.
What do you need to have happen to get that sample? Like, do we need to start phoning you if we find a bat on the island or on the south coast of BC then?
In the winter?
Yeah, I guess so. Like,
well, I don't know, Martin. What do you think? Because the problem is, you see bats in the winter in some of these environments, like caves and mines, but a lot of them end up being Townsend's, which we don't need to really verify. But photos maybe would help to be able to say what species it is, to rule out whether it's worth pursuing. But
certainly are reliable sites. I mean, there's reliable sites that we see them year after year, but they seem to be in the minority. I mean, the number of times I've seen one or two bats in a cave and I go, oh, great, they're hibernating here. And then I go back to sample or whatever, and there's nothing there. It's
and they're gone. Yeah.
Yeah, they're very opportunistic, it seems like, yeah, I'm not sure what it is that drives where they hibernate. I mean, there are consistent sites. You can go back year after year, but they're in the minority, at least on the coast here.
Yes. And this is where I think there are certain areas that maybe we put more emphasis in, like the Northeast, just because there's less species up there. And so the chances of, if 40 kilohertz call, like a recording that's 40 kilohertz up in that area, is almost certain to be either northern myotis or little brown. It's not completely impossible it's long-legged, but I think there's a lot less chance that it would be. And so maybe it's worth putting the time and effort and money into trying to explore some of those areas. But I realize, caving in the winter is really difficult in these remote areas. So it's not like, it's not like I'm pushing to go do that, but if anyone really wanted to, I mean, I think that would be maybe a worthwhile venture at this point. Like the other thing would be to start, I guess just not cavers per se, but for researchers, I guess, to start putting transmitters on bats and late in the fall and starting to try to figure out where they're going. So something like, in some of the areas like Skaha, Kirk, for example, I mean, to start putting some transmitters on bats late in the season, ones you're catching really late, and then see where they end up. I mean, they've got to be using rock crevices, but again, it would be nice to start pointing at the places and say, yeah, that species used this kind of crevice, because we don't really have that information. And maybe we just start with the bats that we can get and start to document that, but hopefully arrive at something for the federally endangered ones eventually. Those are the harder ones.
Mhm.
At this point.
I was going to say winter caving's fine. It's the driest time. It just depends on the access.
The what? Sorry, can you say that again?
I said winter caving can be great because the caves are drier, but it just depends on the access.
Yeah.
To the cave.
Yeah. Yeah, and and it is small numbers. right? I mean, unlike Alberta, BC, yeah, as you said, Martin, it's just like in Kirk. I mean, these are small numbers of bats. So is it even worth pursuing this? I mean, maybe it's nice to be able to do it just at least to verify that we do have some critical habitat. But beyond that, I don't know that it's terribly beneficial to go through and find, you know, a half dozen caves that contain little brown, because they're going to be 1 or 2 little browns and they're probably going to be males. And is there a benefit to having found that? Or do we focus on trying to just get a better sense of winter ecology of the bats that are using the rock crevices, because now we can actually start to predict or model where those bats might be overwintering and other threats that might be posed to them, like winter logging, for example, or you know, I don't know, recreational climbing. I don't know. I'm just thinking about all of these things that maybe we need to understand better about bats in winter, other than identifying caves. But I personally think, you know, it would be nice to at least nail this down a little bit, to be able to point to a few and say that we found those endangered species, and then widen our widen what we want to do in terms of the other 13 species that are overwintering in the province.
Mhm.
Oh, yeah, I was going to ask you, this is a little bit off topic, but is there any more new information about Mexican free-tailed bats in BC?
Excuse me. No, not really. I mean, we've documented them in a few more places, all still in the Southwest. So like Texada Island and more around like Cowichan, in addition to the Salt Spring, and then in kind of that Vancouver area, like, I mean, there's actually been a few of those now, because I have a student that's been biking throughout the summer doing bike transect. She has a bat detector on her bike, and so she's detected a few, and so they're there still, but yeah, nothing, no body in hand, no genetic sample, nothing yet.
Nobody's netted one yet in BC, or
That's what, sorry?
nobody's netted one here?
Oh.
I guess that's a tough one, because they're high flyers, aren't they?
They are high flyers, but and at this point they're just so diffuse. We don't have other than Salt Spring. So Salt Spring, if we were going to try catching them, it seems like that would be the place. But now, see, with eDNA, and I know this had been proposed at one point a few years ago, and I think Peter Omensman and possibly Orville Dyer had proposed something like this. I remember trying to do eDNA on some of the water in Salt Spring Island and see if they could pick up the genetic signature of Brazilian free-tailed, of the Tidarida. But yeah, nothing really came of that I know of. But now that there's so much, is doing a good job of detecting bats. I don't think there's a primer. There's no primers to look for that species necessarily, but that could be something that could be prioritized. Perhaps someone would be interested in developing a primer for that species. And then that's another way to get, yeah, catching it could be hard. I mean, everybody just keeps hoping that one ends up being by a cat, dying somehow. And because 6 bodies showed up in Idaho, and that's how they knew that the species was in Idaho. But yeah, nothing yet in BC, or Washington for that matter.
One other question, the Bats of BC book, when's the new one coming out? And is it going to be a physical book or a PDF, or?
It's a physical book.
Oh, good.
Yeah, old school book. And it has all the updated range maps, but the updated range maps should be appearing on the CDC website too in the new year. So those will be digital.
Okay, so when are they publishing the physical book then?
End of March. Yeah, and WCS Canada, in conjunction with BC Bat Action Team, is hosting the Western Bat Working Group Conference. That happens every 2 years. And this this one this past year was actually virtual because it was supposed to be in Victoria, but it's virtual because of the pandemic. So we are looking now at 2023, in April of 2023, having that conference in Victoria, first time ever in Canada. And we're probably going to have it kind of near the museum and possibly try to promote the book a little or something. I don't know. We're trying to figure that out. But anyways, if anyone wants to come to the BAT conference, April 2023. Sorry, I didn't actually look at the chat, but oh, that's good. No, there's no questions there. Okay. Oh, Ken.
Cori, maybe one other question.
Yep.
These days with COVID, of course, the media is full of conversations about something called herd immunity. And I wonder if after the decimation of a lot of the eastern populations where this thing first manifested itself, is it just a universal picture of continued decline, or have any of the post-collapse population stabilized or even started to recover with the amount of elapsed time?
Yeah, for PD.
Yeah.
Yep, they have actually. So there are a few areas now that were hit really hard where there are bats showing up again. These survivors are literally survivors. You know, they were trying to figure out for a while there, are they just bats that are coming in from outside areas and, you know, filling that void? But there actually are survivors, and the survivors, even though we're talking like small numbers, they're presenting something which allows them to survive. And so, as you can imagine, people would love to study those, and I think for good reason. You know, it's hard to necessarily get a permit to go in and study those survivors. But what looks like so far, the results suggest there's kind of two things happening. Well, actually kind of three. So one is the surviving bats are choosing cooler places to roost. So if they choose these cooler roosts, they've made this behavioral shift that they're in these cooler spots. Or maybe these are the bats that were always choosing to roost in a cooler spot. So they didn't die. But then also they're bigger bats. So the bats that are larger have survived. So there's this shift we're seeing in the population to a behavioral shift to colder roosting conditions in the winter. Larger bats, the more fat, the more likely they're going to survive. And then also there's been a little bit on the adapt genetic component, and it does look like the survivors have shifted in terms of genetics for some of the loci that are associated with hibernation. So there is actually this genetic evolution that does seem to have happened because of this bottleneck that PD caused.
That's really good to hear in today's climate of hopelessness wherever you look. It's really, even though they have a very slow breeding rate, so inevitably, recovery to the pre-collapse populations will take a very long time. At least there's some reason to hope that it might be possible, at least in some places. Yeah,
no, exactly. There's been extirpations for sure, but not extinctions. And so I think, at this point, even though we've got like way more species in the West, almost double the number of species, again, I think the optimistic viewpoint now is, extirpations, declines, severe declines in some of the high visible species, but hopefully no extinctions.
Thank you.
When you say larger bats, do you mean within one species?
Yeah. So little browns in particular. So so a form sort of scales with mass, and so kind of these larger bats that are capable of putting on more fat are being selected for, because bats, because they fly, they're really limited in the amount of fat that they can carry, and so these fatter bats and physically larger bats are more likely to survive within a species. It's like natural selection. right in front of our eyes.
Has there been much work done in Europe and across Asia regarding, where PD has been endemic for a long time in terms of their strategies? I guess they don't have the same number of bats there, so maybe it's kept their numbers down, but has there been much research done into that, how they dealt with it over
There was
there?
early on, there were quite a few papers put out. Pushmail, remember, he was kind of a frontier biologist in kind of looking at what was happening in Europe. And kind of where they ended up landing, as I recall, is mostly, there's a lot less bats. It might have to do with the fact that there was a bottleneck from white nose syndrome 10,000 years ago. So there's that might have actually played into the reason that there were so relatively few bats in Europe. But they also, I've seen a really cool map where they overlaid all of the microclimates that North American bats used to hibernate. They sort of overlaid onto Europe where bats would be hibernating if they were using those same conditions, those nice, cool, moist conditions. And bats in Europe don't use those conditions. So you'd actually see the bats hibernating in Europe are outside of the conditions that North American bats hibernate in. And so again, the hypothesis there is that bats who were using those really great hibernation conditions where the fungus grew well, they died out from this fungus. And so bats of today, you don't see them in hibernating in those conditions for the most part. And so that's what we might see in North America once this is finally wiped through, is where we used to see bats, where we used to think about, this microclimate is what we tend to associate with bat hibernation, will have shifted. And the bats that we now see will be in these cooler and probably drier conditions like they are in Europe.
That's something I've never really heard about, like large colonies of hibernating bats in Europe. I don't know, you probably know more about it than I do, but I've never heard of that over there.
No, again, that's right. Not big groups. Might also be indicative of big groups were killed off by white nose syndrome. Yeah. So they say now, like, in Europe, they don't have white nose syndrome. Well, no, because their bats are sort of living with this fungus, and they've evolved over time, just the way ours are starting to evolve. But there is a little low level, and there has been a paper done that did actually describe there is some still remnant mortality of white nose syndrome in European bats. It's small because a lot of the bats just aren't in a position to be infected anymore. So can I ask you guys a question?
Sure.
So it triggered, trying to see who it was that asked that. I think it might have been you, Jason, but someone asked about, spreading spores. And, here, obviously, we don't necessarily have the fungus in the West, but that's not to say that you guys don't cave elsewhere. But I'm just wondering, like, if you do go from an infected cave, like in the East or something, and then you were to come to the West, like, is there a certain procedure that you guys are following on your equipment? Like, because Martin, you put together a little video and put it on the BatCaver site a few years ago. And was that mostly hot water decontamination? There was quite a few options, but of course you guys don't want to put a lot of chemicals on your gear.
Yeah, that was all the hot water thing. There wasn't really anything else unless you're spraying down equipment. But that was more what we were doing for our own research. So
Yeah, but for a while, the protocols were saying that if you went caving in Tennessee or something, you just don't use that gear back here, period.
Mhm.
I had to buy a new caving suit because of that after the last NSS convention.
Yeah.
I had some cavers visit from the UK and then another couple come from Poland. That was in 2019, I think. And they both brought brand new gear to use in Canada here. So they didn't even want to take the chance.
Yeah. No, that's really good. And that's what I kind of figured was probably happening. Because it is risky. I think if we, if we think about the fact that the spores aren't everywhere yet.
Yeah, but you say they can live for 20 weeks. I mean, that's a pretty substantial amount of time. I mean, you could easily transmit those.
Yeah, and that was I think a little bit of the noteworthy news coming out of that research by Lewis Campbell was that, the assumption was that there's these temperature thresholds, like 20 degrees Celsius over a few weeks should render these spores non-viable. That was sort of the thought for the longest time based on just lab experiments. But now the new research looking at these different scenarios of spores existing on bats, for example, that's where they've realized that maybe those temperature thresholds were a little underestimated, and these spores actually can survive and still be viable for longer at these higher temperatures. But it's still nothing like 50 degrees. And I know that was like what the protocol is for heating stuff up and keeping it immersed in really hot water for 15 or 20 minutes. I mean, that's still, those temperatures are well above what PD can survive. So all the decontamination protocols hold. It's not like that's changed any of those protocols, but I think it's just helping people understand the higher risk associated with bat-to-bat transfer, because we, as bat biologists, we've been kind of thinking in the summer, these bats don't have these spores. They pretty much just have the spores in the winter and the spring when they're really moving around, but that may not be the case. And of course, the bridges has been a real eye-opener for us, because these bats night roost under bridges. They, out in the prairies, it's like one of the few places where they can go. So like somewhere in Saskatchewan, they'd be out like foraging for a good chunk of the night, and then it gets kind of cool and they're kind of full, and they all go hang out under a bridge and digest. And if the PD spores are sitting there, which now we know they are at some of these bridges, then they're pretty much always got this source of PD spore infection that could be happening. And so capturing a bat out in the prairies in the middle of the summer, you would think you're not going to run into PD spores, but you in fact can. And so that's been a big eye opener for a lot of us. Charlene, do you have a bat project figured out yet?
I'm just gonna check to see. Miles unmuted there. Did you have something, Miles? Because you could go ahead.
I'm a little curious about, I heard tell that UV, apparently the PD spores don't survive well in UV light. And it made me think about like a black light UV light as a decon tool, if that would have any effectiveness at all to, because at Horn Lake Caves, we're disinfecting people's footwear, but not necessarily everyone's jackets and things like that. And if there was some other step we could do to disinfect people's clothing, then, maybe just walking around in the sunlight is helpful, but leading up to the caves, but I don't know if there's something else we could do. I'm all for it.
And now this is where I would actually have to go back and look at the paper on this, because there is something, so there's UVA and UVB lights, right? And help me if anybody remembers this, because it is kind of specific as to how you would render these spores inactive using UV. And I think the trickier part when you're starting to talk about clothing and stuff too is just, the UV has to be directly hitting the spore. So if you had like just, even a little fold in fabric, for example, or something, that wouldn't really work. So it's, it's better at really flat surfaces with good UV exposure, close range, that those sorts of things would work well. And like, for example, bat detectors, like our bat detectors, we can't submerge, and most of them, you can't submerge into hot water. Right, you can't heat them up in an oven, and you can't really, you just can't really dunk them or spray them with fungicide and stuff too. So we've struggled with what to do with bat detectors. And of course, roof loggers, the ones that we were using with BatCaver, was that one exception where you could just dump the whole thing into fungicide or hot water. So that was the beauty about them. But we got all these other detectors that are certainly not that robust. And so people were looking at potentially using little UV ovens, if you will, for things like that. But I don't know that it would be widely used on things like clothing. I just don't know that it would work on that.
Or how long you would have to be in that oven for.
Yeah. Like, please walk through this UV oven. Oh, and but it would be like being in one of those tanning salons, right? You'd be able to give everybody the little goggles. There you go.
Shopping cart.
Everybody goes into a tanning salon for X number of minutes and then can go into the cave. I like it.
Now, we can consider clothing, how porous it is and how small spores are and where they can go. I can't see it really being a practical solution.
Yeah. And I haven't seen it widely used for much, like it works, but only under certain conditions. So I don't think, I don't think it would be wide scale.
Works good for water, like treating water, but it's transparent, so works well.
Right. Yeah, that's right. Yeah. Well, yeah, on the simplest, still the simplest thing and for the least amount of chemicals, obviously, is just the water submersions. But you guys at Horn Lake, you're having them walk through, like their feet go on a bath of fungicide, right? Is that what that is still?
Yeah, so that's still a plan that we're going to be re-implementing once self-guiding returns, if that ever does come around. Right now, we're spraying the bottoms of people's shoes with an isopropyl alcohol 70% solution.
Yeah, that's really nice to be able to use that too, because it's a lot easier to tell a visitor that you're going to spray alcohol on the bottom of their shoes rather than fungicide.
Well, actually, the what they've been using is basically just woolite, and my understanding is that that's just the surfactant benefit. It isn't necessarily a fungicide. It's simply to more completely remove dirt and spores and whatever from footwear. I don't think woolite was ever considered to be a fungicide, was it?
No, and I didn't realize it was woolite. It's been a long time since I've been in on any of that early. I was kind of dragged into the early designs on those, the ideas for Horn Lake, but yeah, I didn't follow everywhere that it ended up. So that is woolite. Okay.
Yeah, because in some senses, the idea of walking through a little bath of woolite is probably less effective than a little dusting of isopropyl alcohol to kill spores, or maybe you have to do both. I don't know.
No, the hard part about the isopropyl is so it's 70% isopropyl, and you actually don't want it more concentrated than that because you actually do need the water as part of the process. But keeping it moist for long enough, is hard. It was just like with with COVID protocols too. It's the same thing, right? People were just spraying with isopropyl and then wiping it down real fast, and then that wasn't enough. It has to actually sit there and be wet for a critical period of time, and that's hard to do with alcohol. Like if it's got enough water in it, it doesn't just evaporate instantly. But I think it's the same issue with anything like spraying alcohol. If it's kind of a windy, like I have the same problem with my mist nets, for example. I can't use isopropyl on those because just getting it onto the nets, it just evaporates too quickly. If I could have a little dish of it and I can soak some of my measuring equipment in, that's not a problem. But spraying it onto mist nets is completely impossible. It just goes poof, gone. So that I do have to use fungicide on.
Yeah, it'd be interesting to know if there's any newer research regarding decontamination, because we've sort of been going off the same advice for a few years now, and I don't know if it's changed. Like the woolite thing, I think that originally came from Mammoth National Park.
Yeah. They were using it. The fungal there is exactly what you said, Phil. It's just a really good way to wash things off. It's going to pull pull dirt and stuff out of boots. And it's, it's probably the better way to go anyways, because if you're just, spraying something on the boots, if there is any kind of mud on them, it's not necessarily going to penetrate through them.
Yeah.
So, yeah.
Well,
Hey,
okay.
Cori, sorry.
No,
Did you have
that's okay. I was just reading the chat. Yeah. Hey, Shirley.
Yeah, hey, it's Charlene. So I don't think this is really a question, maybe more of an observation. And it's something that I was, I don't know, enamored with in the caving community when I first got involved with it. And I was kind of surprised by it. And it's the citizen science component. So it comes out pretty obviously when you talk, when Dr. Ann talks. There's also Pat's work with invertebrates, Christian's work on glacial volcanism, Martin and BatCaver. Then there's photo monitoring. Yeah, I just kind of wanted to give a shout now. The follow up to that is now that I'm back in this crazy school academia environment, I'm even more aware of how incredible that opportunity is for a community to be involved in really cool science opportunities without having to go through academia, first of all. And you can be a part of something that really has a tangible outcome. And you're providing a service that others can't do themselves. It's pretty remarkable. And, it comes up a lot in our class discussions and such, or maybe it doesn't come up a lot. Some of us push the topic a lot, but I guess I'm just more kudos to not only the caving community, but also the researchers who are open to working with the citizen scientists of the caving community, because it's a pretty unique and pretty cool thing going on. So I guess part of that is saying to the cavers, jump on every opportunity you can, because I think this is a unique relationship with science that, you should jump on.
Yeah, I'm glad you brought that up, Charlene, because it's, this whole BatCaver program, there's no way that we could have learned this stuff without you guys. Like, I kind of debated about, I'm not a modeler, so I'm not going to explain all these models because I don't even understand them all. But I wanted to at least kind of show you that the data that you guys have collected across Alberta and BC and into Northwest Territories, all of that data contributed to those models because it allowed underground temperatures to be known, and also in terms of occupancy. So here were caves where no bats were detected, and this is how far in the hobo was into the caves roughly, and we did or did not detect bats. And so that information all got rolled up into similar collected data down in the US, and these models emerged on helping to map out where do we find bats and where would we expect to find bats, and how much good underground habitat is likely available for bats in the West. Whether they're using it or not is a whole other question. But then, just potential survivorship, knowing what the fungus needs to grow. So it was just, an invaluable data set that couldn't have happened, without BatCaver. So really, big thanks to everybody in the caving community. And we are trying to do similar things with the bat box stuff, with the community bat programs, and trying to engage more people. And the BC Community Bat Program is, I don't run that one. I work with that group, and we actually operate the Kootenay Community Bat Program, and then WCS operates the Alberta Community Bat Program. But together, we're all just working really hard on getting these emergence count data set together, where people literally are going out all over the country, hundreds of these spots now where people are going and watching bats and counting them and submitting data. And now there's this national bat project with a post office that's joined WCS in conjunction with Canadian Wildlife Federation, and we're doing this national bat box project. So everybody's trying to do it across the nation. We're getting I think there's 1,000 people who have signed up now. So we're going to learn so much from that too. And again, these data sets completely impossible without citizens. So
Yeah, it's very cool. And both you and Anne show a lot of gratitude for that. So yeah, huge respect for that because it shines through all the time that you guys are very grateful for what's offered to you. So thank you.
Yeah, well, thank you.
Corey, I was just going to ask if there's any new technology in the horizon in terms of roost loggers we can use in caves, because for all those ones that I deployed over the years, I was always really disappointed at how few calls we got. I mean, there was caves I was in that where I saw bats and we weren't picking up calls, and you know,
Yeah.
it was disappointing. I was just wondering if you know of anything on the horizon or anything new that's better.
No, I hear you. I wish there was too. I mean, we never did really nail down what was going on with the roost loggers. It might just be low sensitivity. It might have been, you know, because you could program them, right, to turn on and off to save battery power. And what are the chances that, you know, bats are operating during the time that the detector wasn't on, or was it just low sensitivity? I mean, there have been some firmware bugs noticed that have since been fixed, as you know.
Right.
But again, would those have been wide enough to have impacted our results? I don't know. I mean, I think there's a little bit of a random nature to that. I'm not 100% convinced that the roost loggers always worked the way we thought they were working. But I mean, for the most part, they seem to have worked. But yeah, why didn't we pick up in some of these caves where we knew there were bats and we didn't pick them up?
No, I'm just curious about that because, like, for example, I'm still maintaining roost loggers for the Nature Conservancy on Thetis Island, and I put them close to hibernating bats, and they're Townsends, and they tend to wake up a bit in the winter, and they come and go, and you know, I know that. But yet the number of calls that I get on these things is so minimal. I know Townsend's are going to be a little hard to pick up, but I would have thought I'd get a lot more calls. Although on the other hand, I did pick up myotis there, which is really interesting because I've never seen myotis in those cases.
Okay.
So they're good for something.
And that is, I mean, that's a good point, because part of this is we don't necessarily know what the bats are doing, right? And there are some very quiet echolocating bats. Of course, Townsend's is one of the most notorious for being so darn quiet. You don't really pick them up. But then also, if these bats have been hibernating in those same areas for, you know, years, and in many cases, they're probably even hibernating in the exact same spot. Because that's what we've seen, like, you know, over in Catamount Cave, for example, like, you know, the famous banded bat was kind of always in the same chamber, often in the same spot. And that seems to be the case where these bats are just so philopactor, so have such high fidelity that they might be the point where they've been flying that route for like 20 years. And maybe they're not echolocating much. I mean, that's always that possibility, right? I don't know. They seem, you know, you would think they would have to be echolocating to some extent, but maybe they don't echolocate very loudly because the surfaces are all pretty close. And so maybe they don't get picked up on detectors, maybe.
Well, that's kind of one of my theories, because sometimes I'd see bats fly by me and I have a detector in my hand and I wouldn't be picking up anything. And it's like, how are they getting through this and around corners? I thought, well, maybe they're just hearing the echoes from their wing flaps, you know? And
it's highly possible they are cueing in on something else. You're very right, because especially the long-eared bats, because they're used to listening for prey-generated sounds anyways. And so they could be like Myotis evotis, or even like, you know, sometimes we don't think of as long-eared, but like California Myotis is very long-eared relative to its body, and they listen for prey-generated sounds. They're gleaning. So if they can listen for the you know, the slight movement of a spider's foot or the slight flutter of a moth's wing, if they can hear that, then you're right. When they're moving through flapping, they might be able to take advantage of reflected sounds that aren't necessarily ultrasound that they're giving off out of their mouth. I mean, it'd be an interesting study.
Yeah. My strategy in placing the roost loggers was also to find the place of maximum constriction they have to go through, or a tight corner or something like that, because they're more likely to call there. And I'd figure that out just from sitting there where the bat detectors, bats are flying by. So where are they calling at? You know, and they fly up big passages and nothing at all. You don't hear a peep, but then they come up to a constriction and then they'll send out a couple of calls.
And then they do. So. Yep. And that makes a lot of sense. So that might be playing into some of the non-detections, but you're right, and I do remember seeing that in your protocol was trying to, you know, tell people, try to put the detectors at a place where a bat might make a turn. It makes a lot of sense, just in case.
Yeah, but your own science might disregard that, because if they are using bat wing sounds, maybe in a constricted passage, maybe they're getting that bat wing sound more. I don't know, maybe.
That, that's true. So that's what we can do. We can do a study in a cave to try and figure out when do these bats echolocate. And the sad part is it's probably different in different species, and it's possibly, I mean, it's going to be different in every cave too. And anyhow, lots of variables. But I apologize. I'm eating up all of you guys' like cave talk time. If you want any alone time, feel free to say so.
Well, this has been really awesome tonight, Cori. Yeah, huge gratitude for giving your time and your knowledge to us like this. And you did have a deadline in the morning you were trying to race off to. So kind of in reverse, we're sucking up your time. So feel free to run as soon as you need. I would definitely offer that because you've been sticking around a long time.
Oh no, it's okay. I mean, it's been fun, and you guys have had a lot of great questions. It's been a good discussion, and I must say that was something that I did hope we could talk about, was just throwing around ideas about what to do about bat hibernacula and caves, and what, how much further should we push to try to understand what's happening in BC. And because it's something I struggle with, and Martin knows I've been struggling with this for a couple of years actually, like where do we go next with BatCaver? Because if we do start to try to identify just some particular caves that are being used by some of these species at risk, we're going to sink a lot of money into that. And is that where we really want to spend the money? And is it even possible, and is there interest amongst the caving community, because obviously we as bat biologists, well, Martin obviously the exception here, are not cavers as well. So to tackle this sort of a problem involves being able to kind of be a caver and be able to handle the bats too, because we actually would physically have to touch the bat and get a sample from it. And so obviously there's a limited number of you that are in that position. Some over in Alberta, and but
that sounds like a lot of research ethics boards training to me.
Yeah, exactly. So it's which is why there's been all these issues, like how do we move forward with BatCaver?
Yeah.
What can we really do, and what should we do? Where should our priorities lie? I mean, up to this point, like I said, the data that's been gathered through BatCaver has been extremely valuable, and so overall the program's been a great success, and including then over in Alberta, having actually identified some fairly substantial hibernacula too. So big kudos to all of that. But yeah, from this point on, it is a little fuzzy to me as to where we should put our. So if you guys have any thoughts, feel free to reach out to me anyways. Okay, well, maybe I will jump off, get back to my proofs, and yeah, thanks for the invite again. So
thanks so much for coming. It's been most informative, I can tell you.
Oh, that's great. Well, yeah, and we will probably do this again at some point. Feel free to reach out, and and
thank you.
We'll be in touch.
Yeah.
Okay,
Awesome,
thanks,
Cori.
guys.
Thanks.
Thank you.
All right.
Thanks, Cori,
Thanks, bye.
for your time. It was great.
Awesome meeting. I'm going to jump off too. So thank you, Jerry, for putting this together, and have a good.
Yeah, likely me too. Good to see everybody. Even though you didn't see me.
I haven't had dinner yet. I think it's
Take care, everyone. We can shut it down.
Yeah, that was a
now.
great night. Great.
Yeah, it's okay.